Good morning, everybody. We're going to be talking about mitosis, which is cell division, cells coming from other cells, being created by other cells. So if you go back to our beginning of the year, we talked about the cell theory, and one of the key components of the cell theory is that all cells arise from pre-existing cells through the process of cell division, which is mitosis, meaning that cells don't just randomly appear. Cells come from a pre-existing cell. A cell is going to divide into two cells, or sometimes it's a smaller organism grows off the side of a larger organism. It's not just going to come from nothing, it's going to come from other cells. So why do cells divide? We have a few different reasons. First one, reproduction. We can see here this small single-celled organism is making a copy of itself. It's dividing into two cells, and this is how that species uses to divide. There's also growth, which is where over the course of an organism's lifetime, it begins to increase in size. We can see a human here going from a baby over to a human. Sorry, I mean an adult human. Again, your cells aren't increasing in size, it's the number of cells that's increasing. That's how we go from this beginning of the phase to the larger size over the course of the lifetime. There's also repair. If you're injured, it's going to heal. It's going to create scar tissue and over time leave possibly a scar depending on how it heals. But that's going to be, and closing up wounds as well, stopping bleeding is going to be part of the repair process. And development. So we're constantly growing and developing, even though we might be like, if I stop growing in terms of stature and height, it doesn't mean that my body continues to, or stop developing. Right now, your bodies are developing, your organs are still developing, your neural pathways, meaning your brain is still developing, and that's why it's important to make sure that you stay healthy and active, especially during this course of your lifetime, because your bodies are continuing to develop and grow. So mitosis is the division of somatic cells. So somatic cells are non-reproductive body cells. Reproductive cells, in contrast, would be sperm and egg cells, cells that are used to make a zygote, which eventually would grow into a baby and then eventually give birth and you'd have a human. So somatic cells are non-reproductive cells, perform mitosis in our bodies. So it develops into two identical daughter cells. Now, daughter cells have the same number of chromosomes as the parent cell. So these two daughter cells down here are going to be identical to this parent cell all the way over here. So the first phase, or not really the first phase of mitosis, what the cell spends most of its time in, is cell existing, is called interphase. So interphase is, we can see here in this diagram here, this is the cell cycle as we call it. This large portion of the cell's life, the majority of its life, is going to be spent in interphase. A very small portion is over here in M phase, which is what mitosis is, which is the dividing stage. As you can see here, we end up with two daughter cells. So what is interphase? Interphase is the non-dividing stage, basically the cell existing, doing whatever its function in our bodies, continuing to help us maintain homeostasis. It's not a stage of mitosis. So a cell is preparing for mitosis during interphase. And what does it do to prepare? It's beginning to grow in size. It swells a little bit. And then the nuclear membrane, the nucleus, the nuclear envelope, is going to disintegrate. But that's, again, what's inside of it? The DNA. The DNA is now going to be exposed, which is helpful. And the most important part during this interphase for mitosis is that the DNA is replicated, DNA replication, which is meaning that we're making a copy of it. So what is DNA? We talked about DNA a few times this year. It's a nucleic acid, deoxyribonucleic acid. In the form where it's unfurled, it basically looks like a loose ball of string. That's going to be chromatin. That's uncondensed DNA molecules. So being uncondensed is important because it makes it easier for our bodies using enzymes and ribosomes to make copies of it. It's going to be able to duplicate it because each new daughter cell is going to need a copy of that DNA because, again, they're going to be genetically identical to the parent. The first phase of mitosis is called prophase. So again, interphase is not a phase of mitosis. It's the cell just existing, getting ready for mitosis. Mitosis, the actual dividing process. The first phase is prophase. So in prophase, what we're going to see is a few things. We're going to see that DNA, chromatin, that unwound ball of string, is going to condense into chromosomes. So the DNA has been replicated in interphase, and now it's condensing into chromosomes. So this makes the cell division a lot easier. So chromosomes are made of an identical pair of sister chromatids attached by a centromere. So here's what that looks like. So here we have a chromatid, which is one copy of the DNA. So we have a centromere at the center. When we have two together, we have two sister chromatids together. This creates a chromosome. These are also referred to as chromosomes, but for our purposes, we're just going to refer to this attached for right now during this phase of the process as the chromosome. Two sister chromatids, which again, if I were to draw a line down the middle, they're two identical copies of each other, are going to be given. One goes to one daughter cell, one goes to the other during a later phase of mitosis. So spindle fibers are also beginning to form. So spindle fibers, as we learned in another video, are microtubules. They're going to be very, as it says in the name, micro, small tubules, tubes, very small tubes that we also call spindle fibers. And they're going to radiate out from these centrosomes. Centrosomes, as you can see here, are made of two parts. These two orange pieces, those are centrioles. So centrosomes are made of two centrioles. So the spindle fibers begin to form. And the next phase shows why that's important. Prometaphase, which is the second phase, is going to be where those spindle fibers begin to attach to the chromosomes. They begin to specifically attach to the centromeres of the chromosomes. So that center part where the two chromatids are attached to each other. This leads us into metaphase, a very important phase. If this phase does not go correctly, mitosis actually stops. The entire process is canceled. So the chromosomes are going to align at the equator of the cell. They form what's called the metaphase plate. So this straight line of chromosomes down the center, we have our centrosomes on either side with the spindle fibers attaching. When the chromosomes line up at the center, as we see here, which is an actual picture through a microscope, that's going to be called the metaphase plate. If the metaphase plate doesn't properly form, it's not going to work. An easy way, or a little trick that I like to use to remember which phase is which, because we have to remember the right order of them. Metaphase, think middle. M for metaphase, M for middle. That's going to help you remember that they line up along the middle during this specific phase. Anaphase is where we're going to start seeing how the DNA is divided between the two cells. This is where the sister chromatids are separated as they're pulled away towards the centrioles, again, which are centrosomes. So we can see here the X's are gone because we split them in two. We broke them into two separate halves. We can see this picture clearly illustrates it over here, that the chromosomes are being separated, so the chromatids are being pulled towards opposite ends of the cell. This picture is kind of cool. It looks like two separate jellyfish. So the legs of these, again, not actual jellyfish, but the ones that looks like the legs of a jellyfish are actually the spindle fibers pulling the pulling the chromatids towards the centrosomes. Telophase is where we begin to have the spindle fibers disintegrate because the process is coming to an end. We're going to be beginning to get ready to split into two separate daughter cells. The nuclear envelopes around both groups of chromosome begin to form. So we're creating a new nucleus around the DNA. And the chromosomes revert to their extended form, meaning they're uncondensing. So they go from a ball of string, like a loose ball of string, to chromatids or chromosomes, which is where they're really condensed. They have that X shape. And then once they are divided back into their two separate daughter cells, once they're found, we have two identical daughter cells, they uncondense again because DNA is much easier to work with when it's uncondensed. Next is going to be where we have cytokinesis, which is the actual creating of two separate daughter cells. So the cells are going to split apart. And remember, the DNA inside these two nuclei are identical to each other because they're coming from the same parent cell, which, as we saw in interphase, made a copy of itself. So the overall process we see here, we have prophase, which is where, again, just to reiterate, that's where the DNA condenses into chromosomes, spindle fibers begin to form. Prometaphase, which is where we have the spindle fibers attached to the chromosomes. Metaphase, which is where the chromosomes line along the middle. Remember, metaphase, middle. Anaphase, the chromatids are split apart. Telophase, they begin to reform the nucleus. The spindle fibers begin to fall apart. Cytokinesis/cleavage is going to be where the cells split, and then the cells, as we see here, are going to go about their existence. So the last thing I want to talk about today is the difference between diploid versus haploid cells. This is a term that we're going to see sometimes also pronounced diploid. Some people pronounce diploid, some people pronounce it diploid. This is going to be important for not only mitosis, but we're going to see later in meiosis and also in genetics. So a diploid cell is a cell that contains two sets of chromosomes. One set is donated from each parent. So your biological mother and your biological father each gave you a set of chromosomes. We have in our species 46. So that means we got 23 from mom and 23 from dad. So they are called homologous pairs. So they carry the same genes. When I say genes, again, we're going to go into this in a little bit in the future. When I say genes, I'm not talking about like the same exact version of a gene. So let's say there's an eye color gene. They carry. So let's say a certain chromosome carries the eye color gene. That might not be the same version of a gene. Those are called alleles. Alleles. So you could have the allele for brown eyes, or you can have the allele for blue eyes, or the allele for green eyes. There's going to be many different versions of the same gene, and those are called alleles. To show it that a cell is diploid, we use the symbol 2n to represent the diploid chromosome number. Because again, we have n is the standard number of, or the number of different chromosomes, and 2 showing that we have one from each pair. So we have we have two separate sets. These are the non-reproductive cells, the somatic cells, the body cells. These are the white blood cells, the neurons, the skin cells. This is what are going to be diploid cells. Haploid is going to be the cells that only have one set of chromosomes. So these are represented by just a singular N because they only have one set. They only have 23 chromosomes. Diploid for our species has 46. Haploid has 23. So reproductive cells such as sperm and egg cells would be haploid. So think about it. If let's say that the sperm cell has 23, the egg cell has 23. When you put them together, you have 46 to make a diploid cell. And when you have sperm and egg come together, we'll see this in a little bit, that's going to be a zygote, which is eventually going to grow and develop into a baby. So here is, if we were looking at mitosis in terms of diploid, this is what the process would look like. We start off with a diploid parent cell, and that's going to divide, represented by the two red arrows here. Those represent the process of mitosis: prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis, and then we end up with two daughter cells. And not only are they identical in terms of they have the same number of genes, but they have identical genes. Is there a chance that a random mutation can occur? Yeah, absolutely. But it's very, very minimal compared to the number of actual bases that make up our DNA to have an actual mutation that will have an effect on the daughter cells. So again, the parent is going to divide and form two identical daughter cells. So all three cells in this picture, the two daughter cells when compared to each other or comparing the daughter cells to the parent, are identical genetically to each other. Okay, I will be in contact with all of you soon, and I look forward to hearing from you. Bye, everybody.